Bismillahirrahmanirrahim. Assalamu alaikum. In the previous lecture, we have started with the topic of control of respiration. In today's lecture, we are going to continue with the same topic. In the last lecture, we talked in detail about the respiratory control center present in the brain stem. And this respiratory control center is for automatic control of respiration. Although the respiratory activity is automatic and it is generated by the centers, by the neurons present in the brain stem, however, the activity of the neurons in the brain stem, it is modified in response to various inputs to the respiratory center. So there are different stimuli, there are different sensory inputs which are transmitted to the respiratory center, and these sensory inputs in turn modify the activity of the respiratory control center. In today's lecture, we are going to talk about the inputs to the respiratory center. There are different types of inputs, a large variety of inputs to the respiratory center. We are going to see the different factors that can modify the respiratory activity, and we will focus our discussion on the peripheral and the central chemoreceptors in today's lecture. Here we can see different inputs to the respiratory center. This is the respiratory control center present in the brain stem, and it receives inputs from multiple areas, from the periphery as well as from the higher areas of the brain. In this diagram, the positive sign indicate a stimulatory effect on the respiratory center in the brain stem, which means these inputs are going to increase the activity of neurons in the respiratory center. And negative sign indicates an inhibitory influence on the respiratory center, which decreases the activity of the respiratory control center, mainly the dorsal respiratory group of neurons. So respiratory center, it receives signals from the higher areas of the brain, that is the cerebral cortex, and these signals mainly are to mediate the voluntary control of breathing, and these signals can be stimulatory as well as inhibitory. In the last lecture, I have told you that we can have a partial control of our breathing by hyperventilating for a short period of time or by holding the breath for a short period of time. So if we are hyperventilating, so this is a stimulatory effect, and if we are holding the breath, this is an inhibitory influence of the cerebral cortex on the respiratory center. Then respiratory center also receives signals from the hypothalamus, and these signals basically mediate the effect of pain and different emotional states on the respiratory center. So again, in these situations, there can be either a stimulatory effect on the respiratory center resulting in increased respiratory activity, or there can be inhibition of the or you can say can be a decrease in the respiratory activity. Then the chemical stimuli can also produce their effect on the respiratory center. The chemical stimuli in the blood, they produce their effect by stimulation of either the peripheral chemoreceptors or the central chemoreceptors. In this aspect, the three chemical stimuli are very important. These include a decreased oxygen in the blood, that is the arterial blood. So decreased partial pressure of oxygen in the arterial blood, increased partial pressure of carbon dioxide in the arterial blood, and increased hydrogen ions. These can mediate their effect either through the peripheral chemoreceptors or through the central chemoreceptors. The central chemoreceptors are present within the brain, and the signals which are transmitted from the peripheral and the central chemoreceptors to the respiratory center, these have a stimulatory effect on the activity of the respiratory control center. Then the proprioceptors are present in the muscles and joints. These proprioceptors are stimulated by the movements of the joints, and the signals are sent from these proprioceptors to the respiratory control center, and again these have a stimulatory effect. In the lungs, we have irritant receptors. These irritant receptors, when are stimulated, they have an inhibitory influence to modify the respiratory activity. Lungs also have stretch receptors, and when stretch receptors are stimulated, they also have an effect of decreasing the activity of the neurons in the respiratory control center. And all these inputs to the respiratory control center, they mainly are transmitted to the dorsal respiratory group of neurons, the neurons which are present in the nucleus of tractus solitarius. So in today's lecture, we will mainly talk about the peripheral chemoreceptors and central chemoreceptors, and in the next lecture we will talk about the other inputs to the respiratory control center. Now, the control of respiration through changes in the concentration of partial pressure of oxygen in the blood, or the concentration of carbon dioxide and hydrogen ions in the blood, is termed as the chemical control of respiration. We can say that there are two types of control of respiration: the nervous control that we have discussed in the previous lecture, and the other control is chemical control, which we are going to discuss in today's lecture in detail. And for the chemical control of respiration, the stimuli are the changes in the concentration of oxygen, carbon dioxide and hydrogen ions in the arterial blood. Decreased partial pressure of oxygen is going to stimulate the respiration. Increase in the concentration of carbon dioxide and increased concentration of hydrogen ions stimulate the respiration. For increased concentration of hydrogen ions, we can also say a fall in the pH of arterial blood. So one thing you must remember, that changes in the concentration of these chemicals in the arterial blood is going to have a profound effect on the respiration. So we are not concerned with the changes in the concentration in the venous blood. Changes in the concentration of these chemicals in the arterial blood is going to have a profound effect on the respiration. Now these stimuli, they produce their effect through stimulation of chemical receptors, and these chemical receptors are termed as chemoreceptors. Now chemoreceptors, as we also discussed in the previous slide, are of two types: the central chemoreceptors and the peripheral chemoreceptors. Now we are going to talk in detail about both types of chemoreceptors, their stimuli, the mechanism of stimulation, and the effect of stimulation of these chemoreceptors on the respiratory control center. First, we are going to talk about the central chemoreceptors. Now, central chemoreceptors are also known as medullary chemosensitive area. This is very important to remember, that this chemosensitive area or the central chemoreceptors, these are distinct. This is a separate area from the respiratory center that we have discussed in the previous lecture. So this is a separate area present in the brain stem, 0.2 millimeter beneath the ventral surface of medulla. So So this is the location of the central chemoreceptors. I am repeating that central chemoreceptors are present in the medulla about 0.2 millimeter beneath its ventral surface, and this area is separate from the respiratory center itself. Now what is the stimulus for the excitation of the central chemoreceptors? The primary stimulus for the activation of central chemoreceptors is a fall in the pH of interstitial fluid and CSF. You can also say that increase in concentration of hydrogen ions in the interstitial fluid and the CSF is a primary, potent, direct stimulus for the central chemoreceptors. However, this increased concentration of hydrogen ions or a fall in the pH of interstitial fluid and CSF, it is brought about by an increased concentration of carbon dioxide in the arterial blood. This is very important. You must remember that increased concentration of carbon dioxide in the arterial blood leads to a secondary increase in the concentration of hydrogen ions in the interstitial fluid of the brain and in the CSF, which in turn stimulates the central chemoreceptors. Now, how does carbon dioxide increases the concentration of hydrogen ions? We will see in detail shortly. Now, we can say that the pH of the interstitial fluid is regulated by the partial pressure of carbon dioxide in the arterial blood, because it influences the concentration of hydrogen ions in the interstitial fluid. If there is an increase in the partial pressure of carbon dioxide in the arterial blood, it will diffuse into the interstitium of the brain and into the CSF, because it is lipid soluble and it can easily cross the blood-brain barrier. In the interstitium of the brain and in the CSF, it will lead to increased formation of hydrogen ions, which is a potent direct stimulus for the central chemoreceptors. Another very important point to remember and understand is that an increase in hydrogen ions in the arterial blood does not stimulate the central chemoreceptors, because hydrogen ions in the arterial blood cannot cross the blood-brain barrier. Now look at this diagram. This shows us the location of the central chemoreceptors. As I just told you that the central chemoreceptors is a distinct area present 0.2 millimeter beneath the ventral surface of medulla. So this area is the chemosensitive area or the central chemoreceptors. When the neurons in the central chemoreceptors are excited, they will send signals to the inspiratory area of the respiratory center, and we know from our previous discussion that this inspiratory area is the dorsal respiratory group of neurons. Now, when is this chemosensitive area stimulated or excited? This area is stimulated, or we can say the neurons in the chemosensitive area are excited when there is increase in the concentration of hydrogen ions in the interstitium and in the CSF. Increase in the concentration of hydrogen ions. जब hydrogen ions ज़्यादा हो जाएंगे, increased concentration will stimulate the neurons in the chemosensitive area. Now, how these hydrogen ions are going to be increased? These hydrogen ions are increased as a result of increase in the partial pressure of carbon dioxide in the arterial blood. Now look at this diagram. This diagram shows us the central chemoreceptors. You can see here, these are the central chemoreceptors, and these are the respiratory control center present in the brainstem. Whenever there is an increased partial pressure of carbon dioxide in the blood, it will lead to an increased concentration of carbon dioxide in the interstitium and in the CSF. So this is CSF. Now here in carbon dioxide is going to react with water. This will result in formation of carbonic acid. Now this carbonic acid, it dissociates into hydrogen ions and the bicarbonate ions. Now these hydrogen ions which are formed here, now these hydrogen ions are going to stimulate the central chemoreceptors, and when central chemoreceptors are excited, they will send signal to the inspiratory area or to the dorsal respiratory group of neurons, which will increase their excitation, and this will result in an increase in ventilation. So the hydrogen ions are produced within the CSF and within the interstitium. However, the cause of increase in concentration of hydrogen ions is an increase in the partial pressure of carbon dioxide in the arterial blood. So that is how the central chemoreceptors are stimulated, and you can see that the hydrogen ions which are present in the blood, they cannot cross this barrier, which is the blood-brain barrier, a barrier present between the blood and the brain. Hydrogen ions cannot cross this blood-brain barrier. So the increase in the hydrogen ions in the blood will not have an effect on the central chemoreceptors. Arterial hydrogen ions, when these are increased, they cannot stimulate the central chemoreceptors because they cannot cross the blood-brain barrier. So this is the mechanism of stimulation of central chemoreceptors and their effect on the pulmonary ventilation. Again the diagram to show you the same. This is the blood-brain barrier. Carbon dioxide is lipid soluble. It can cross the blood-brain barrier and it locally produces the hydrogen ions within the CSF and in the interstitium, which stimulates the central chemoreceptors. But hydrogen ions in the arterial blood cannot cross this blood-brain barrier. Therefore, these hydrogen ions have no influence on the central chemoreceptors. So we can conclude that central chemoreceptors are stimulated by increase in the blood carbon dioxide. Whenever there is increased concentration of carbon dioxide in the blood, this is going to stimulate the central chemoreceptors through formation of hydrogen ions. And hydrogen ions in the CSF and in the interstitium of the brain is going to stimulate the central chemoreceptors. While central chemoreceptors are insensitive to the hydrogen ions present in the blood, and these are also not stimulated by the oxygen concentration, changes in oxygen concentration in the blood. Now, this is very important to remember that changes in the concentration of carbon dioxide, that is the increase in concentration of carbon dioxide, it has an acute effect on controlling the respiratory activity. That is, when there is increased concentration of carbon dioxide in the blood, this will stimulate the respiratory center, and this effect will stay there for about 1 to 2 days. So the effect of carbon dioxide to cause an increased respiratory activity is an acute effect lasting for 1 to 2 days. It has a weak effect for chronic control of respiration. a weak chronic effect. Now, this is because of the fact that in chronic conditions, which means that when carbon dioxide is chronically increased in the arterial blood, the central chemoreceptors lose their sensitivity to changes in arterial PCO2. Now, this loss of sensitivity of the central chemoreceptors is because of the adjustment of the hydrogen ion concentration in the blood by the kidneys. Now, whenever there is increased concentration of carbon dioxide in the blood, this will cause a fall in the pH of the blood, and this will cause the kidneys to compensate for it. Now, kidneys will compensate for it by increasing the excretion of hydrogen ions and increasing the reabsorption of bicarbonate ions, which will bring the pH back to normal, and the central chemoreceptors will lose their sensitivity to increased partial pressure of carbon dioxide in the arterial blood. So, acute effect of carbon dioxide in controlling the respiratory activity, and it has a weak chronic effect. Now, this graph shows us the effect of increased partial pressure of carbon dioxide and of a fall in the pH on the rate of pulmonary ventilation. So, whenever there is increased partial pressure of carbon dioxide, it has a very potent acute effect in increasing the rate of alveolar ventilation, while a fall in the pH of the blood or an increase in the hydrogen ion concentration in the arterial blood also increases the rate of alveolar ventilation, but this is not through the stimulation of central chemoreceptors. How does increased hydrogen ions in the arterial blood affect the pulmonary ventilation? We are going to see subsequently. Now, decreased partial pressure of oxygen does not stimulate the central chemoreceptors. We have discussed shortly before. Now, this decreased partial pressure of oxygen, it reflexly increases the respiration through stimulation of the peripheral chemoreceptors. So, in the beginning we have talked that there are two types of chemoreceptors, the central and the peripheral chemoreceptors. So, we can conclude so far that central chemoreceptors are stimulated by increase in the partial pressure of carbon dioxide in the arterial blood, and we have seen the mechanism in detail. How does the increased partial pressure of carbon dioxide has an influence on stimulation of the central chemoreceptors. So now we are going to talk about the peripheral chemoreceptors, which are stimulated by the decreased partial pressure of oxygen in the arterial blood. Now this is a primary and a potent stimulus for the peripheral chemoreceptors, that is a fall in the partial pressure of oxygen in the arterial blood. It strongly stimulates the peripheral chemoreceptors. When the partial pressure of oxygen decreases, it falls below 60 millimeter of mercury. Now this fall in the partial pressure of oxygen in the arterial blood is not the only stimulus for the peripheral chemoreceptors. And remember that peripheral chemoreceptors are also stimulated by increase in partial pressure of carbon dioxide in the arterial blood, and these are also stimulated by a fall in the pH of the arterial blood. So partial pressure of carbon dioxide, when it is increased, it not only stimulates the central chemoreceptors, but it also stimulates the peripheral chemoreceptors, while a fall in the pH of the arterial blood or an increase in the hydrogen ion concentration in the arterial blood, it produces its effects on the respiration through stimulation of peripheral chemoreceptors, because the arterial hydrogens cannot cross the blood-brain barrier, so they cannot enter the brain and they cannot stimulate the central chemoreceptors, and these hydrogen ions produce their effect through peripheral chemoreceptors. Now we are going to discuss the peripheral chemoreceptors in detail, especially in reference to the decrease in partial pressure of oxygen in the arterial blood. First, we are going to see what are the peripheral chemoreceptors. Now, peripheral chemoreceptors are the chemoreceptors present in two important locations in the body, that are the aortic and the carotid bodies. Aortic bodies are present in the arch of aorta, and the carotid bodies are present at the bifurcation of the common carotid artery. Now, the aortic and the carotid bodies have specialized cells which are known as glomus cells. Now, these glomus cells, along with their sensory nerve endings, form the peripheral chemoreceptors. I am repeating that peripheral chemoreceptors present in the aortic and in the carotid bodies, they have specialized cells which are known as glomus cells. These glomus cells, in association with their sensory nerve endings, form the peripheral chemoreceptors. And the potent stimulus for the peripheral chemoreceptors is a fall in the partial pressure of oxygen. Look at this diagram. It shows us the location of the chemoreceptors that are the peripheral chemoreceptors. So we have aortic chemoreceptors present in the arch of aorta. These are present in the aortic bodies. And the carotid chemoreceptors are present in the carotid bodies, which are present in the bifurcation of the common carotid arteries. Now, this graph shows us the effect of a fall in the partial pressure of oxygen in the arterial blood on the rate of stimulation of the carotid chemoreceptors. So, at 100 millimeter of mercury, there is the rate of discharge of impulses by the carotid chemoreceptors is very low. When partial pressure of oxygen decreases below 100, in the range of 60, when it falls below 60 to 30, there is potent stimulation of the carotid chemoreceptors, and the rate of discharge of impulses by these chemoreceptors is tremendously increased. So this graph shows us a potent stimulation of the chemoreceptors, that are the peripheral chemoreceptors, when there is a fall in the partial pressure of oxygen below 60 millimeter of mercury. Again, it shows the effect of a fall in the partial pressure of oxygen on the rate of alveolar ventilation. In the previous graph, we have seen the effect of fall of partial pressure of oxygen on the stimulation of the carotid receptors. And when these carotid chemoreceptors are stimulated, they in turn send signals to the respiratory center, which in turn increases the rate of alveolar ventilation. So when there is a fall in the partial pressure of oxygen in the arterial blood, there is increase in the rate of alveolar ventilation, and this potent increase is seen when partial pressure it falls below 60 millimeter of mercury. And this shows that in this situation, this effect of decreased partial pressure of oxygen on alveolar ventilation rate is studied while keeping the partial pressure of carbon dioxide in the arterial blood to a constant level. Now, when peripheral chemoreceptors are excited, they send sensory signals to the respiratory control centers present in the brain. Their signals are transmitted via the glossopharyngeal and the vagus nerve. Now these nerve signals through the glossopharyngeal and the vagus nerve terminate in the nucleus of tractus solitarius, which constitute the dorsal respiratory group of neurons, which is the inspiratory area present in the respiratory center. So these are the aortic chemoreceptors. The excitation of these receptors cause generation of the nerve signals, which are transmitted to the brain stem through the vagus nerve. So signals from aortic chemoreceptors are transmitted to the respiratory center through vagus nerve, and the signals from the carotid chemoreceptors are transmitted through the hearing nerves to the glossopharyngeal, and through the glossopharyngeal nerve to the respiratory center present in the brain stem. Now we are going to learn the mechanism of stimulation of the peripheral chemoreceptors. Mechanism of stimulation of peripheral chemoreceptors when there is a fall in the partial pressure of oxygen in the arterial blood. When there is a fall in the partial pressure of oxygen in the arterial blood, this is sensed by the glomus cells. As I just told you that glomus cells are specialized cells present in the aortic and the carotid chemoreceptors. Now this causes, whenever there is decrease in the partial pressure of oxygen in the arterial blood, this causes the closure of oxygen sensitive potassium channels present in the membranes of the glomus cell. Now due to closure of the oxygen sensitive potassium channels, potassium stays inside the cell. It does not diffuse out from the cell. It stays within the cell, due to which there is increased concentration of positive ion inside the cells, leading to depolarization. Increased potassium within the cell causes depolarization of the cell, and this depolarization in turn causes the opening of the voltage gated calcium channels. Closure of potassium channels, which are oxygen sensitive potassium channels, causes depolarization because more potassium is accumulated within the cell, and this in turn will lead to opening of the voltage gated calcium channel. Opening of the voltage gated calcium channel will lead to influx of calcium into the cells. Now due to influx of calcium into the cells, there is release of the neurotransmitters, because the neurotransmitter vesicles are going to fuse with the cell membrane, and the neurotransmitter is released by process of exocytosis. It is believed that acetylcholine and ATP act as neurotransmitter. Now the neurotransmitter which is released, it will bind with the receptors present in the afferent nerve fibers that innervate these receptors. And I just have told you that in case of aortic chemoreceptors, these are innervated by the vagus nerve. Their nerve fibers, they are transmitted to the brainstem through the vagus nerve. And in case of carotid chemoreceptors, the nerve fibers present in the glossopharyngeal nerve are going to innervate the carotid chemoreceptors. So this will lead to excitation of the afferent nerve fibers. Binding of the neurotransmitters with the receptors will cause excitation of the afferent nerve fibers. They will generate the nerve signals which are transmitted to the brain stem, to the respiratory control center, mainly the nucleus of tractus solitarius. Again, I am repeating that from aortic chemoreceptors, the signals are transmitted through the vagus nerve, and from the carotid chemoreceptors, the signals are transmitted through the glossopharyngeal nerve. And in the beginning of the lecture, we have discussed that chemoreceptors, they have a stimulatory effect on the respiratory center. So this will result in an increased pulmonary ventilation. So decreased partial pressure of oxygen in the arterial blood will stimulate the peripheral chemoreceptors, and this will send signals to the brain stem, and the result is increase in pulmonary ventilation. And this increase in pulmonary ventilation is brought about by increase in tidal volume first, and then by increase in respiratory rate. So there is increase both in the tidal volume and the respiratory rate, which will cause an increase in pulmonary ventilation. Now this diagram shows us the summary of the effect of different chemicals in the blood on the respiratory activity through the peripheral and the central chemoreceptors. So we talked about the chemicals in the arterial blood. So if there is a decrease in the partial pressure of oxygen in the arterial blood, and especially if it falls below 60 millimeter of mercury, this is going to cause a potent stimulation of the peripheral chemoreceptors. Peripheral chemoreceptors are excited, nerve signals are generated and are transmitted through the vagus and the glossopharyngeal nerve to the respiratory center. It has a stimulatory effect on the respiratory center to cause increase in ventilation through increase in the tidal volume and increase in the respiratory rate. If there is an increase in the partial pressure of carbon dioxide in the arterial blood, it is going to stimulate the peripheral chemoreceptors as well as it is going to stimulate the central chemoreceptors. So increase in partial pressure of carbon dioxide, it has effect on stimulation of peripheral as well as the central chemoreceptors. Through peripheral chemoreceptors, the signals are transmitted to the brain stem to result in increase in pulmonary ventilation. And increased partial pressure of carbon dioxide, it also increases the concentration of carbon dioxide in the CSF and in the interstitium, because it is lipid soluble and it can cross the blood-brain barrier. And within the brain, it causes increased concentration of hydrogen ions to the reactions that we have already discussed, and this increased hydrogen ions will have a potent stimulatory effect on the central chemoreceptors. And when the central chemoreceptors are excited, the signals are transmitted to the respiratory control areas. Again, these are stimulatory signals, and this will increase the activity of the dorsal respiratory group of neurons to increase the pulmonary ventilation. The third chemical which can influence the respiratory activity is increased hydrogen ions in the arterial blood, or a fall in the pH of arterial blood. Now this will have a stimulatory effect on the respiration or on the ventilation through the peripheral chemoreceptors. So whenever there is a fall in the pH of arterial blood or there is an increase in the hydrogen ion concentration, this will stimulate the peripheral chemoreceptors, and signals from the peripheral chemoreceptors are transmitted to the respiratory control centers, and this will increase the pulmonary ventilation. And remember that these hydrogen ions do not stimulate the central chemoreceptors. Hydrogen ions in the arterial blood do not stimulate the central chemoreceptors because these cannot cross the blood-brain barrier, and instead they produce their effect through stimulation of peripheral chemoreceptors. So again the same thing, effect of changes in the concentration of oxygen, hydrogen and carbon dioxide on the respiratory center, and all these have a stimulatory effect. A fall in the partial pressure of oxygen an increase in the hydrogen ions, and increase in the partial pressure of carbon dioxide. Again, I am repeating that increased partial pressure of carbon dioxide produces the effect through stimulation of both the central and the peripheral chemoreceptors. It can directly stimulate the peripheral chemoreceptors, but it stimulates the central chemoreceptors by formation of hydrogen ions, while arterial hydrogen ions stimulate the peripheral chemoreceptors, and decreased partial pressure of oxygen in the blood also stimulates the peripheral chemoreceptors. These two parameters, that is decreased partial pressure of oxygen in the arterial blood and increased hydrogen ions in the arterial blood, have no effect on the central chemoreceptors. So that's all for today's lecture. These are some points that you must remember, that the peripheral and central inputs are integrated in the nervous system. Pulmonary ventilation is very sensitive to changes in PCO2, which acts through the central chemoreceptors mainly, but it can also stimulate the peripheral chemoreceptors. And remember that day-to-day regulation of respiration, it is brought about by the stimulation of central chemoreceptors through changes in the concentration of carbon dioxide within the physiological limits, while the peripheral chemoreceptors, they become an important drive for the respiration in situations which are beyond the physiological limits. That is, when there is decreased partial pressure of partial pressure of oxygen in the arterial blood, there is hypoxia under abnormal situations. This will lead to the stimulation of the peripheral chemoreceptors, and in these situations, in chronic conditions, the peripheral chemoreceptors have the role in regulation of respiration, and in acute condition and in day-to-day regulation of the respiration, PCO2 is important. So this I have told you, that under physiological and pathological conditions associated with hypoxia, peripheral chemoreceptors have the significant role. So that's all for today. I hope you understand the lecture, and if you have a problem in understanding any of the concept given in today's lecture, you are free to ask question. Thank you for the listening.